Welcome to the review on large-scale chromosome aberrations. Problem 1. Plant species A has a diploid chromosome number 2n equals 10. Species B has diploid number 2n equals 20, and species C has a diploid number 2n equals 30. Based on this information, determine if the following statements are true or false. Statement A. An allotetraploid involving species A and C has a total chromosome number of 40. So remember, allotetraploid means that it's a tetraploid which has 4 complete sets of chromosomes, and allo means that 2 species or more are involved. If it was autotetraploid, that would mean that only 1 species is involved. So you're doing an allotetraploid, so what you're looking at would be the combination of 2 diploids. So species A has a diploid number of 10, and species C has a diploid number of 30, so an allotetraploid of them together would equal 40 chromosomes. So that's true. Statement B. A gamete from an autotetraploid from species B has a total of 30 chromosomes. So remember what I just explained. Tetraploid has 4 complete sets of chromosomes, and auto means that it comes from the same species. So if a diploid species B has a number of 20, that means that an autotetraploid would have a tetraploid number of 40. So let's write that. So 4n would equal 40. And a gamete from that autotetraploid would be half, right? Because that's what meiosis does. It reduces the chromosome number in half. So half of that would be 20. So this is false. And to make it true, this would have to be 20. Okay? So for statement C, an autotetraploid for species A has a total chromosome number of 40. Again, they're telling us in the problem that a diploid number 2n equals 10, so the tetraploid number, which would be 4n, would be twice that, so that would equal 20. So this statement is false, and this should be 20. Okay? Part D. A monosomic individual from species A has 9 chromosomes. Well, this is true. Remember that monosomy can be written as 2n minus 1 because you're missing 1 chromosome out of a homologous pair. So for plant species A, the diploid number is 10, so that's 2n, and 10 minus 1 would equal 9, so that's true. Statement E. A trisomic individual from species C has 33 chromosomes. So this is actually false. So trisomy can be written as 2n plus 1 because there's a diploid organism has 2 copies of every chromosome, and trisomy means you actually have 3 copies of 1 chromosome, so you're adding an extra copy. So if you look at species C, the diploid number is 30, and therefore the trisomy number would be 31, not 33. Okay? So this is a mix of aneuploidy and polyploidy, but it's important to know how to write out chromosome contents where you do 2n minus 1, 2n plus 1, etc. To solve these types of problems. Problem 2. A haploid yeast strain carrying 2 linked recessive markers, His17 and Lys2, is auxotrophic for histidine and lysine. The strain is crossed and sporulated with a wild-type strain, dominant His17 and dominant Lys2, that has an inversion in this region shown in blue. So, as you can see as the inversion region shown in blue, this is a pericentric inversion, which means that it includes the centromere. For each of the following crossover events that could occur in the diploid during meiosis, determine the genotype and phenotype of the 4 resulting spores. Assume that any deficiencies in genetic material are lethal in haploid spores. Do not consider crossovers between sister chromatids. So again, as we did for the paracentric inversion, remember that when a diploid is heterozygous for inversion, where 1 chromosome is not inverted and the other is, you're going to get what's known as an inversion loop when they line up for during meiosis. So let me draw that here. So we have the first one. Remember, you draw as a bubble, the center of which is the centromere, and you have recessive histidine, recessive lysine. Then you have the second chromosome, which forms the loop. it has its centromere in the middle. And then you have dominant histidine, dominant lysine. Okay, so the first crossover event that we're going to do is a single crossover outside of the inverted region. So that would be right here. It could have also been on the other side, but it really doesn't matter here. So let's do the highlighting to follow along where these go. Now remember, what I drew here, just as we did earlier in the first section of this chapter, was that I only drew 2 out of the 4 chromosomes that are really lining up at this point in meiosis. So remember that each of these chromosomes has an exact sister chromatid attached to it, but it would just be too messy to draw, and also they're telling us not to consider any crossovers between the sister chromatids. So remember again that the 2 that we don't show are going to give us our parental phenotypic spores. Okay, so let's do this first crossover. We'll start on the left top, follow the crossover down, follow the loop, you get dominant lysine, dominant histidine, and there you go. Now the other, you follow from the bottom chromosome up, you get recessive histidine, recessive lysine. Okay, so those are our 2 recombinants. So for A, you get 4 spores, 2 of which are parental. So you get P1 and P2. Now the parental, remember, are going to be recessive histidine, recessive lysine, which means auxotrophic for both. That's the phenotype. The other will be dominant histidine, dominant lysine, prototrophic for both. And then you have the 2 recombinants. So this first recombinant in yellow gives you dominant lysine, dominant histidine, prototrophic. And the one in green, the recombinant in green, gives you recessive for both, which means auxotrophic for both. So these look parental with their phenotype, but they were generated through recombination. Okay? And all 4 are viable, and they all have the parental phenotype. So let me draw this again so we can do part B. You have the 2 recessive markers. And then you have the 2 dominant markers. Now for this, I'm going to do something a little bit extra for this one. What we want to do is put selective markers at the end. Okay? When we did the paracentric inversion problem in the previous section, we didn't have to do any outside markers because we had the centromeres on the ends, right? But this time, in order to see if these spores will be viable, you need to put flanking markers to see. Okay? So on this top chromosome, let's put big A on the left, big B on the right. And on this bottom chromosome, let's put little a on the left, little b on the right. So now the crossover event that's occurring is a single crossover between the dominant His marker and the centromere. So the crossover that they want would be somewhere in here. So let's take our highlighter and go across and see what we get. So the first one, let's start at the big dominant A marker. You follow along the chromosome, you get recessive His, follow down the crossing over, you get dominant Lys, and then you come around and you get recessive A. Now for the other one, we're going to have to start on the right, so we get dominant B, recessive lysine, then you follow the crossover down, you get dominant histidine, you follow through, and you get recessive B. So what are these recombinants that we're getting here? R1, the one in yellow, we get big A, little histidine, dominant lysine, and then little a. And then R2, we get big B, recessive lysine, dominant histidine, and little b. So what do these flanking markers tell you? Well, this recombinant 1, this spore, has a duplication in gene A, and it's missing gene B. Now remember in the question, it says, assume that any deficiencies in genetic material are lethal in the haploid spore. So now these recombinants that we're talking about are spores. And it's saying that any deficiency is lethal. So for R1, that's deficient in B, so this is lethal. And R2 is deficient in A, which is also lethal. So the recombinants here are, neither of them are viable, so they're both lethal. So the only spores you'll get are the first parent, which is auxotrophic for both, and the second parent, which is prototrophic for both. Okay, again, I'm going to write this one more time, the inversion loop, to do part C. Okay, and I'll draw markers on this one too, just in case it becomes a problem. The reason why I didn't draw it on 1, if you see if I did, just to show, you did draw markers here. You get dominant A on the yellow chromosome, leading to recessive B. The green chromosome recombinant would have recessive A with dominant B. So you wouldn't be deficient, it would just be rather if you have the dominant or recessive would switch. Okay, so for C now it's asking a double crossover event where one crossover is between histidine and the centromere and the other is between lysine and the centromere. Assume that both crossovers involve the same 2 chromatids. So again, it doesn't matter that we didn't draw the sister chromatids, all that's occurring is with these 2 that we drew. Okay, so the first crossover is between his and the centromere, which would be here, and the second crossover between the centromere and the lysine marker. Okay, so let's take our highlighters and follow through the crossover events. So first you start with a dominant A, then you get recessive histidine, you drop down to this crossover, get through the centromere, come back up, now you get recessive lysine, and dominant B. Okay, then you get little a, go up, you get dominant lysine, then you follow the crossover up, get a centromere, follow it back down and get dominant histidine, and then you follow to get recessive B. So if you see, what actually happened was the 2 chromosomes actually just switched centromeres. So the yellow chromosome still has dominant A, recessive his and lys, and dominant B, but it just switched centromeres with the green chromosomes. So let me write down the spores for here. You have P1 and P2, just like normal, the parentals. Then you have R1, which is A, recessive his, lys, dominant B, which is auxotrophic for his and lys. So trophic. And then you have recombinant 2, which is little a, dominant his, dominant lys, little b, auxotrophic for his and lys. And again, remember that these look parental, but are generated through recombination. Okay, so when you're doing pericentric inversions, as we did here, remember that it's smart to draw the markers outside, because when you do pericentric, the centromere on the end serves as a marker. So you, as we saw, we had dicentric and acentric, but here you need to draw these outside markers to see if they'll be lethal, like in part B. Problem 3. A diagram of the gene sequence of a normal chromosome is shown below. Determine which chromosome mutation is illustrated by the following chromosomes. So what I did, I took each of the subparts of this and put it on a different page, so that on each page we can see the normal chromosome above and then the mutated chromosome below, so that way we can compare them and see what mutation actually happened. So for part A, when you're comparing the normal chromosome gene sequence to the mutated one, you can just go from left to right. You see you have Z, A, G, Y, and then you're missing this X, right here. So I'm just going to circle where you see the mutation. Then you have the centromere, and then you have CE. So the only mutation, the only change from this mutated chromosome from the original was missing X. So this is a deletion of gene X, right? And a deletion is where you're missing a piece of the chromosome, and in this case, it did contain a gene. For part B, again, you have the normal chromosome above and the mutated chromosome below. If you go from left to right, you see ZA, and then instead of being ZYX, as in the normal chromosome, you see you have XYG on the mutated chromosome. And then you have the centromere and then CE. So this switching, if you can tell, instead of being GYX, it just flipped it or inverted it to XYG. So this is an inversion of GYX. And to get more specific, it's actually a paracentric inversion because it does not contain the centromere in the inverted portion. So paracentric, no centromere involved. Part C, let's compare again. You have ZA, but then if you look, there's an extra ZA in this mutated chromosome, right? And then if you continue, you see GYX centromere CE. So in this mutation, there's actually an extra piece of chromosome that contains the G, Z, and A. So this is a duplication of Z and A. And this is also what's known as a tandem duplication, because the duplication happens one right after the other. If ZA was duplicated and then was put, let's say, down here, then that would just be a regular duplication. But because these are next to each other, we call this a tandem duplication. And finally, part D, you have ZAG, Y, but then you have, on the top, you have X centromere CE, and that's, remember, this is the normal chromosome. But on the mutated chromosome, you have EC centromere X. So this is the part that was mutated, and as you can see, again, this is just a flipping or an inversion. So this is an inversion of XCE, but in this case, it does include the centromere, where here there was two genes at the end after the centromere, C and E. In the mutated chromosome, there's only one gene, and that's because the centromere was flipped and inverted along with those other pieces. So this is called a pericentric inversion. And that's because it contains the centromere. Okay, so this completes this problem, and these were just some visual illustrations of what these mutations can look like when you put genes on a chromosome. Problem 4. A species has a chromosome content characterized by 2n equals 18. How many chromosomes will be found per cell in each of the following mutants? So we know that this species is a diploid with a chromosome number of 18. So we also know that the haploid number here, which would be n, equals 9 chromosomes. So this should be a good review of how each of these chromosome aberrations play out in terms of chromosome content. Okay, so part A says for a monosomic cell, how many chromosomes would you expect? So remember that monosomy is when one chromosome of a chromosome pair is missing. So this is characterized by the formula 2n minus 1, because remember in a diploid, all of the chromosomes have a pair. Each chromosome set is in 2. So monosomy is where you're just missing one of one pair. So 2n minus 1 would be 17 chromosomes. B says tetrasomic. So this is where an organism has 4 copies, that's what the tetra comes from, is 4, of one chromosome. So remember again, in a diploid, organisms generally have 2 of one chromosome. So in a tetrasomic individual, they would have 2n plus 2, because to have 4 of one chromosome, you would need 2 extra. So 2n plus 2 would be 20 chromosomes. C, an autotriploid. Remember, when auto, that means it's coming from the same species, which makes sense because they didn't give us any chromosome number for another species. And triploidy is a type of euploidy where it's an addition or a subtraction of a complete set of chromosomes. So triploidy is where you have 3 complete sets of chromosomes. So diploids have 2 complete sets, and this part C, this mutant has 3 complete sets. So this is characterized as 3N. And to find this, you want to take the haploid number 9 and times it by 3, which would give you 27 chromosomes. Part D is autopentaploid. Again, the auto means that all the chromosomes are coming from this species, and a pentaploid means that they have 5 complete sets of these chromosomes. So again, you take the haploid number, which is 9, multiply it by 5, and that gives you 45 chromosomes. E is a trisomic. So this is where it's a type of aneuploidy, where it's not a complete set of chromosomes, but however, you just have 3 copies of one specific chromosome. So again, remember, diploids have 2 copies of each chromosome, so to get another one, you would have to do 2n plus 1, so that equals 19 chromosomes. F is a double monosomic. So remember from part A, monosomy is where you're missing 1 out of the 2 copies of a chromosome. In a double monosomy, that means, let's say you have, well, they have 18 chromosomes, right? Pairs. So that means that you're missing 1 of the chromosomes from pair 1, and you're missing another chromosome from, let's say, pair 3. So to get a double monosomy, this would be 2n minus 2, because you're missing a chromosome from 2 separate sets. So this would be 16. Okay? G is nullisomy, and this is where 1 chromosome set is completely missing. So if a haploid number is 9 here in this species, that means that there are 9 pairs of chromosomes to give the diploid number 18. Nullisomy is 2n minus 2, because you're missing 1 complete set of chromosomes. So again, that'd be 16. So as you can see, part F and part G, a double monosomy and a nullisomy, gives the same number of chromosomes, but these chromosome contents will be different. Okay? So even though they both have 16 pairs of chromosomes, this G, this nullisomic individual, will have 16 chromosomes that are complete paired, 8 pairs. But in this double monosomy, you will have 6 pairs, which will give you 7 pairs, which will give you 14, and then you'll have 2 extra chromosomes that don't have any pairs, because remember that monosomy is where you lose 1 chromosome from a pair. Okay? Then finally, autotetraploidy. Again, auto means from the same species, and tetra is where 4 comes from, so this would be 4n, and this would give you 36 chromosomes, and this is from chromosome doubling. Okay, and there's some mutagens that Professor Schmidt spoke about in the theory video that can actually lead to this chromosome doubling. So this is a good representation of all the different types of chromosome aberrations you can get and how it will affect the number of chromosomes in each cell. So just be careful again with F and G. Even though they have the same number of chromosomes, they will have different chromosomes in those cells. Number 5. Species 1 has a diploid number of 8 chromosomes, and species 2 has a diploid number of 14 chromosomes. Determine the expected chromosome numbers for individuals with the following chromosome mutations. Give all possible answers. So let's just write out what we have. We know that species 1 is 2n diploid number equals 8, and therefore n haploid number equals 4. And species 2 is 2n equals 14, n equals 7. Okay? So part A says autotetraploidy in species 1. Remember that auto means that all the extra chromosomes are coming from that same species. And tetraploidy means you have 4 complete sets of chromosomes. So in species 1, we can write this as 4n. And in species 1, if you do 4 times the haploid number, which is 4, that will give you 16 chromosomes. So as we said in the last problem, this is what's known as chromosome doubling. Part B, trisomy in species 2. Trisomy is where you have 3 copies of one chromosome. Now in a diploid organism, they already have 2 copies of every chromosome, so the chromosome content can be characterized as 2n plus 1, and that plus 1 gives you that 3rd copy of a chromosome. So 2n plus 1 in species 2 would give you 15 chromosomes. Part C, allotriploidy including species 1 and species 2. Here is where this distinction happens. So it's auto versus allo. Auto means that the chromosomes, the extra chromosome content comes from that specific species. Allo means that the chromosome content is a mix between 2 or more different species. So it's saying here it's including both of the species. So allotriploidy we can write as 3x. Now when it's an alloploidy, we usually write the chromosome content as a number times x instead of a number times n, just to show that these chromosomes are coming from different individuals. Okay, so 3x can have a few possible answers. So let's write that down here. 3x can be from 2n of species 1 and 1n, or haploid, of species 2, or it could be 1n of species 1 and 2n of species 2. Okay, so let's find both of these answers. So if you have 2n of species 1, that would be 8, right, because that's their diploid number, and 1n of species 2 is 7, so that gives you a total of 15 chromosomes. Or you can have 1n from species 1, which equals 4 chromosomes, and 2n from species 2 equals 14 chromosomes, and that gives you a total of 18 chromosomes. Okay, so part C equals 15 or 18, depending on where the 2n comes from, either species 1 or species 2. Part D says monosomy in species 1. Remember that monosomy is where one copy of one chromosome is missing. So in a diploid that has 2 copies of every chromosome, you would do 2n minus 1 to lose that one copy of a chromosome. So species 1, 2n is 8, so 2n minus 1 is going to equal 7. Part E, tetrasomy of species 2. Remember that tetrasomy is where you have 4 copies of one chromosome, and a diploid already has 2 copies, so to get another 2 to get 4, you just add 2. And in species 2, 2n equals 14, so you add 2, and that gives you 16. And now part F, allotetraploidy including species 1 and species 2. Again, remember that allo means that it's coming a fusion of the chromosomes from 2 different species, and the tetraploidy we can write as 4x. Now again, this part has multiple answers, so let's write that below. So how can you get 4x? Well, you can get 2n of species 1 and 2n of species 2. You can get 1n of species 1 and 3n of species 2. Or you can get 3n of species 1 plus 1n of species 2. So let's find out what these equal. So species 1, 2n is 8, and species 2, 2n is 14. So that gives you 22 chromosomes. Or you can have 1n from species 1, which is 4, and 3n of species 2 would be 3 times 7, which is 21. So that gives you a total of 25 chromosomes. Or you can have 3n of species 1, which would be 4 times 3 is 12, plus 1n of species 2 is 7. So that gives you a total of 19 chromosomes. Okay, so F can either be 22 or 25 or 19, depending on where the chromosome content is coming from and how it's split up. Okay, So this is fairly straightforward. Again, just another review of the formula to finding chromosome content, but just be careful with these allotetraploides. And it's involving two species. Remember that there are multiple ways that you can get an alloploidy species. Let's finish up this chapter review by doing these true-false statements. Number 1. Pseudodominance is the phenomenon where a recessive allele is quote-unquote unmasked due to a deletion of a chromosome segment. So this is true, right? This is what a possible phenotypic effect is of a deletion of a chromosome segment. So let me draw an example. Let's say you have the part of a chromosome that encodes for eye color. And in a diploid organism you have two chromosomes. And we'll say that on one chromosome you have the big B allele, which encodes for brown eyes, and on the other you have the little b allele, which encodes for blue eyes. Now, a normal diploid would have the phenotype of brown eyes. However, if you deleted it, let's say you deleted, I'll highlight it instead of erasing so you can look back. Let's say you deleted this part of the chromosome. Now the only allele for eye color that this individual has is this little b allele. So if you deleted the yellow portion, the phenotype of this individual would be blue eyes. So even though in the normal chromosome content, this individual has the dominant allele for brown eyes, with a deletion, this recessive allele for blue eyes is unmasked, and therefore the phenotype changes after a deletion. So this is, not that much of a consequential effect, but let's say if it was a lethal allele, where you had a dominant allele that would make you viable, but the recessive one was lethal, well then if you had the deletion of that dominant allele, you wouldn't be able to survive without, because the lethal allele will be unmasked. Okay, so that's why deletions have a very huge phenotypic effect on an organism. Okay, number 2. Pericentric chromosomal inversions do not include the centromere. This is false. Remember that pericentric chromosome inversions do involve the centromere, and it is paracentric inversions that don't involve the centromere. Number 3. Inversions tend to have more serious phenotypic effects than other chromosomal rearrangements. Well, this is false, and for a reason that we just talked about with deletions can unmask recessive alleles, which is known as pseudodominance, and also inversions can cause a great phenotypic effect because, not inversions, duplications, because you'll have more than the required amount of gene product, and that can cause dosage problems. So let me just write that. Deletions and duplications can cause dosage problems. Okay, and that can lead to a more serious phenotypic effect than inversion. So inversions, remember, just as where you take like a span of 3 genes and you just flip them around so that the third gene is now first, the middle stays the same, and now that last gene is what was first before. So as long as an inversions don't break in between a gene segment, they really don't have that much of an effect because the genes will still be expressed. Number 4. An allotetraploid species is likely to be able to produce viable gametes and may be agriculturally important. So this is true, and I know Professor Schmidt went through a few examples in the theory video that a lot of agriculturally important plants are actually polyploid species and allopolyploid species, and this makes them bigger, it makes them more able to live on the land, and they can produce viable gametes and can reproduce. Number 5. allodiploids are created through the fusion of diploid gametes. So just take a look at this. It says allodiploids. Remember that allo means from two different species, and diploids mean 2N. And it's saying that it's created through the fusion of diploid gametes. So this word right here is what's making this false. So an allodiploid is created by the fusion of normal haploid gametes from different but closely related species. So I'll write it in red here. This word should be haploid gametes in order for it to be correct. And finally, number 6. If a normal diploid cell has the chromosome number 2N, a nullosomic cell has a chromosome content of 2N minus 2. Remember that nullosomy is missing one whole set of chromosomes. So let's say an individual has 4 pairs of chromosomes for a 2N number of 8. If you're missing, let's say, pair 2, that means you only have pair 1, 3, and 4. So you're only going to have 6 chromosomes. So a way to write this is 2N minus 2, because a normal diploid has 2 copies of every chromosome. And if you're missing both copies of one of them, then you're going to subtract 2. So this is true. It is the formula for writing a nullosomic individual that is originally diploid. Okay, so this concludes the chapter on large-scale chromosome aberrations. And if you have any questions, I think what the best way to do is go through, and these formulas like this where it's 2N minus 2, 2N minus 1, 2N plus 1, go through and make sure you know which chromosome aberration gives you each formula, and I think that will really help out.